Have you ever wondered what you really look like on the inside? I mean, really on the inside, at a scale so tiny it feels almost imaginary. Well, today we're going to take a journey into this hidden universe that exists inside every single one of us. It's a world of unbelievable complexity and just stunning beauty.
Take a look at this. You might think it's something you'd see hanging in a modern art gallery, right? But this is not an abstract painting. This is a real, super magnified image of biological tissue. It's this incredible landscape of fibers and cells all working together. And those colors, those patterns, that's all happening inside you right now.
So that's the big question, isn't it? I mean, our own tissues are mostly water. They're super soft, and to our eyes, they're pretty much colorless. Plus, the cells that make them up are microscopic. So how on earth do scientists take that invisible, fragile world and turn it into these amazing, informative images we can actually study?
To answer that, we've got to dive into the science and the art of making the invisible visible.
The field is called histology. It actually comes from two Greek words, histo, which means tissue, and logos, which means study. So, put it together, and it's simply the study of tissues. You can think of it like the instruction manual for how our bodies are built, showing us exactly how all those individual cells get organized into the complex stuff that makes up our organs.
Now, to get a clear picture, histologists have to overcome four huge challenges. First, any piece of tissue is opaque. Light just can't pass through it to get to a microscope lens. Second, as soon as you take tissue out of the body, it's soft and it starts to decay, like immediately. Third, even if you could see it, everything's basically transparent. And finally, of course, it's all way too small to see. So, how do you solve all that?
Well, the first part of the answer is this really clever preparation process, where you turn a soft piece of tissue into an impossibly thin slice.
This diagram here shows the game plan perfectly. You start with this small, delicate tissue sample, and the whole point is to make it stable and firm enough so you can cut it into paper-thin, transparent sections. It's quite a journey from something soft and wet to a solid, stable block.
The process has 4 key steps. First up is fixation. You use a chemical, usually formalin, to lock all the proteins in place and just stop decay dead in its tracks. Next, dehydration. See, water and wax don't mix, so you have to get all the water out, and you do that with a series of alcohol baths. Third is clearing, where you swap out the alcohol for a solvent that can mix with wax, and that leads to the grand finale, embedding. The tissue gets soaked in hot paraffin wax, which seeps into every single nook and cranny. Then, as it cools, the whole thing hardens into a solid block. It's basically like freezing Han Solo in carbonite, but for science.
Okay, so now our tissue is nice and solid. We need to slice it. And for that, you use this thing, the microtome. It's like an ultra-precise deli slicer for science. It can cut that wax block into sections so thin it's almost hard to believe. Just 1 to 10 micrometers. To put that in perspective, that's about 10 times thinner than a single human hair. The slices are so thin, they're completely transparent.
All right, so we've solved the problem of the tissue being too fragile and too thick. But now we have a totally new problem. Our perfect, ultra-thin slice is completely invisible.
Yeah, the slice is basically a ghost sitting on a glass slide. All the important stuff is there, but we just can't see it. So to reveal the story that's hidden in that tissue, we have to add color. This is a process called staining, and it's where the science really starts to feel like art.
And the most common, the absolute rock star of the staining world is H&E. That stands for hematoxylin and eosin. It's the workhorse of literally every histology lab, and it transforms that invisible slice into this vibrant landscape of pinks and purples. Suddenly you can see a ton of detail.
So how does it work? It's actually genius in its simplicity. Hematoxylin is a blue dye, and it likes acidic things in the cell, while the most acidic part is the nucleus, which is packed with DNA. So it stains all the nuclei a deep purple blue. Eosin, on the other hand, is a pink dye that likes the basic parts, like all the proteins in the cytoplasm. So with just 2 colors, you can instantly tell the difference between the cell's command center and its factory floor.
But H&E is just the beginning. Histologists have a whole palette of special stains they can use. If you want to see things like mucus, you use a PAS stain, which turns it bright magenta. To see the tiny fiber network that holds everything together, a reticulin stain makes it pop in black. Or, like in that very first image we saw, there's Mawson's trichrome, which is awesome because it separates collagen in blue from muscle in red. Each stain tells a different piece of the story.
Okay, so our slide is preserved, it's sliced, and it's stained.
It's this perfect little piece of art. But there's one last problem we have to solve. It's still microscopic.
And for that, of course, we need the classic tool of biology, the light microscope. It just shines a beam of light through our specimen, and a series of glass lenses magnify that image, usually up to about 1,000 times its actual size. And just like that, our hidden world finally comes into view.
But there's a limit. There's this thing called resolving power. It's the ability to tell that two objects close together are actually two separate things. Because of the physics of light itself, a normal light microscope just can't resolve anything closer than about 0.2 micrometers. So to see the really tiny machinery inside our cells, we needed a breakthrough. We needed a whole new kind of light. And that breakthrough was the electron microscope. The transmission electron microscope, or TEM, doesn't use light at all. It shoots a beam of electrons through an even thinner slice of the specimen. And because electrons have a much, much shorter wavelength than light, the resolution is just incredible. It lets us peer deep inside a single cell and see all its little parts in amazing detail.
The other main type is the scanning electron microscope, or SEM. Now, instead of passing electrons through the sample, its beam scans back and forth across the surface. And the result isn't a flat 2D slice, but this breathtaking 3D-like image. I mean, look at these blood cells. The difference is just wild. You feel like you could almost reach out and touch them.
So, through all of these steps, preserving, slicing, staining, and magnifying, we finally beat all the challenges. We can finally read the cellular story.
Okay, let's go all the way back to where we started, to that abstract painting. Now we know exactly what we're looking at. This slide was stained with Masson's trichrome, so we know that the brilliant blue stuff is collagen, the main structural protein. The rich red is muscle tissue. And all those tiny dark purple dots, those are the individual nuclei of the cells. See? It's not abstract at all. It's a precise map of life.
And as amazing as these images are to look at, histology isn't just about making biological art. It's one of the most powerful and fundamental tools in all of medicine.
I mean, this field matters because it's how we understand the basic blueprint of our own bodies. It's how doctors diagnose diseases like cancer from a biopsy. It's how pathologists can figure out why an organ failed. They can even freeze a piece of tissue during a surgery, slice it and stain it in just a few minutes, and tell the surgeon right then and there if they've removed a whole tumor. It's incredible stuff.
So in the end, every single one of these slides, these hidden landscapes, it tells a story. It might be a story of perfect health, or it could be the very first clue that helps diagnose a disease. It's the secret language of our bodies, written in the architecture of our cells. It really makes you wonder, doesn't it? What story is hidden inside of you?